As I think about the evolution of gene editing, so we think about sort of CRISPR Cas9, Nobel Award-winning technology that was for the first time with very, very high efficiency, you can edit a very specific place within the DNA. And that was really groundbreaking technology. And what it's really good at is the search function of finding the right place in the DNA that you actually want to cut. And with a CRISPR Cas9 enzyme, you're making a double-stranded break within that DNA. And by making that double-stranded break, there are things that can happen as a result of that. You can have unwanted off-target editing that you prefer not to have, and it can also cause chromosomal translocations, chromosomal rearrangements, other things or sequelae that we call them that you prefer not to have with a gene editing technology. But it is very good at sort of knocking down genes, and there's a number of programs that are obviously in the clinic. Now we have an approved therapy for sickle cell disease. So again, a really important therapy, I think, for patients. And then out of David Liu's lab came another gene editing technology, and this is called base editing. So I'll give you a couple of the examples of sort of what changed. So instead of using that CRISPR Cas9 enzyme that does a double-stranded break, now you're using a modified Cas enzyme. So you're making a single-stranded break. And by doing that, you don't get a lot of the off-target effects that you might get with CRISPR-Cas9 editing. But you do get something that's called bystander edits in some cases. So in not every case are you taking a patient back to sort of normal or what we call wild-type protein. And then out of David Liu's lab came something called prime editing. So we're adding sort of a different enzyme to the mix. We're still using that modified Cas enzyme, so we have those single-stranded breaks. And I like to think about it as we've got the most versatile way of editing your genome, and I think it's the safest way to edit the genome. So in terms of versatility, we can make any type of edit. So we can do what CRISPR-Cas9 does. We can knock things out. We can do what base editing does. It's just changing one base pair to another. So it's sort of limited to 4 base pairs where they can do that. And then we can do all of that and then so much more. So there's a lot that CRISPR-Cas9 and base editing can't do that we can then go and target with prime editing. That includes things like we can excise repeat expansions, we can fix transversion mutations, we can fix frame shift mutations, and with our passage technology, we can actually insert very large amounts of DNA. So it's just an incredibly versatile approach to gene editing. And then both, again, from a safety standpoint, we actually use CRISPR-Cas9 as sort of our positive control in our experiments, and we really don't see any of these off-target effects, translocations, rearrangements, or bystander edits that you would get with other types of technologies. So it's a really powerful tool.